So this presentation is about DNA cycle sequencing. So what is it? Well, DNA cycle sequencing is basically a way to determine the order of specific nucleotides on a section of DNA that you want. So if you want to find out the nucleotides of a virus or something, you could use it. So step one. Step one of DNA cycle sequencing is going to be obtain all the materials you're going to need. So water, dNTPs, ddNTPs, primers specific to the DNA, Taqpolymerase, and the template DNA. And what you're going to do is just put it all in the test tube and mix it up. Now, step two. Step two is going to be to heat up the test tube with all the materials in it using a thermocycler to 96 degrees Celsius, almost boiling in fact. You're going to want to do this so that you can start denaturation. So, what is denaturation? Denaturation is when water is heated up to a very high temperature, in this case 96 degrees Celsius, until the hydrogen bonds between the nucleotides weaken until they break. This breaks apart the DNA into two single strands. Now, step 3. Step 3 is when the test tube is cooled down to 55 degrees Celsius, which is still very hot, 131 degrees Fahrenheit, so that primer annealing can start. The temperature can fluctuate though if there are a lot of cytosine or guanine bases. What is primer annealing? Well, primer annealing is when the test tube is cooled down so that the primers you put in the test tube earlier can bind to that template DNA. But unlike PCR, only one primer attaches to each strand of DNA. Step 4. Step 4 is, once the primers have attached to the section of DNA that you want to replicate, extension starts to occur. What is extension? Well, extension is when the Taqpolymerase, which is a kind of polymerase that can live in very high temperatures, starts to add surrounding dNTPs and ddNTPs to the corresponding DNA strand that the primer started. Step 5. Step 5 is, as extension is occurring, it's adding nucleotides, but sometimes it adds what is called ddNTPs, and the strand stops there. That is because the ddNTPs have no hydroxide bond, so other dNTPs cannot attach. Step 6. Step 6 is just to repeat steps 2 through 5, which is denaturation, primer annealing, and extension, and you just repeat this until there are millions of copies of that original template DNA of all different lengths because of that ddNTP. Step 7. Step 7 is to put your newly created DNA through gel electrophoresis, which sorts it by length, and put that newly sorted DNA through a different machine that detects its ddNTPs because of its bright glow. This is one of the reasons that DNA cycle sequencing only uses one primer. If two primers were used, when it runs through the machine would scan not only the ddNTP, but the corresponding dNTP. So then the information would be hard to read. Step 8. Step 8 is when the information from the machine you just put the DNA through is transferred onto a long piece of paper to then be analyzed. The information on this piece of paper are the individual dNTPs that the machine detected.